And our grand rounds today is occurring in the context of an event that we've suffered this past week with the passing of Roselle and Tara Borelli. And when news of this occurred over the weekend, and we plan to have that departmental meeting on Monday, thought about whether we should hold grand rounds or should we use that as the forum for the town hall grieving session, but first we had tried for some time to get Damien Fair to be our speaker and had arranged it, so we didn't want to sort of press our luck. And then in addition to that, Rosellen would have wanted us to get back to business as usual. And so that's what we're going to do. Just to say, in addition, that we're talking to the family about posting an obituary if that's what they would like us to do. Lou Baptista is leading a planning session for a memorial session. And there are still available for any faculty or staff the resources through COPE Columbia that Lou, Amy, Claude, and Laurel have been orchestrating. And there's a website. You just go to COPE Columbia at cumccolumbia.edu, or there's a direct line for individual counseling, 646-774-6311. So with that, I'd like to turn the platform over to Guillermo Jorge to introduce our Grand Round speaker. Guillermo.
Hi, everyone. I'm thrilled to have Damien Fair as our Grand Round speaker today, who comes to us from the University of Minnesota, where he recently moved. Before then, Dr. Fair completed his neuroscience PhD training at WashU under the primary guidance of Bradley Schleger and Steve Peterson. He then completed postdoctoral training at Oregon Health and Science University with Joel Nigg and Bonnie Nagel, where he spent 10 years as principal investigator of the Developmental Cognition and Neuroimaging Labs and a champion for diversity, equity, and inclusion efforts. Dr. Fair recently co-founded the Masonic Institute for the Developing Brain, where he is the Red Leaf Endowed Director, and became professor in the School of Medicine at the University of Minnesota. Damien is a world-renowned leader in the fields of developmental and cognitive clinical neuroscience, and he develops and uses advanced functional and structural MRI methods to study brain development in typical and atypical populations, including ADHD and autism. Has published more than 100 articles in the top research journals, including Science, Nature, Neuroscience, Neuron, and PNAS, just to name a few. And his research has been funded by grants from the Gates Foundation, McDonnell Foundation, and NIH. Has also received many awards for his work, including the Presidential Early Career Award for Scientists and Engineers, issued by President Barack Obama in the White House, and most recently, the MacArthur Genius Award. Today, he will tell us about his lab's recent work on developmental cognitive neuroscience in the era of big data. So with that, I would like to give a warm welcome to Dr. Damien Fair. Damien.
All right, thank you. Thank you very much for having me. I'm really sorry to hear about your colleague. I hope everybody's doing okay with that. But I am definitely very happy to speak to you. It's been kind of a long road of making this go, so it's great to be here and to present some of the ways we're thinking about how to do our research these days. Let me get my screen shared here. I think this should be working. Okay. So, all right. So what I will start with here is just I always like to start. It's a very busy slide to begin with, in part because I always like to let people recognize that it really, like, the number of people that have gone into and collaborated on the work that we've been doing is just absolutely enormous. We have folks from all different types of disciplines, obviously. Clinical practitioners, engineers, obviously neuroscientists, physicists. I mean, it's just an enormous amount of effort that requires lots of different types of expertise to carry out the work. And so I always like to make that point up front, just in case I don't get to do a good full acknowledgement at the end. Now, I'm going to have three main goals of today's talk. One is I'll describe, what we're calling the ABCD BIDS Community Collection, collection 3164. It's a new collection that I'll be describing about community participation with the ABCD study. I'll describe we call the replication problem in our field, and others is now at a very specific turning point that I think is critical, that we really think deeply about how we're doing our science. And then I'll end with a bit of what we're kind of doing related to what are we going to do about it, increasing and improving the SNR in some of our measurements, and kind of leveraging individualized network topology using the ABCD data as well. Okay, so many of you here are familiar with the ABCD study, but I'll just kind of give just a brief overview. So this is a study that was started about 5 years ago, a little over 5 years ago, with an aim of across 21 different sites of collecting 10,000 9 and 10-year-olds and following them over the next 10 years, collecting all different types of measurements and scanning them every other year to look at developmental trajectories, particularly during periods of adolescence where lots of experimentation and lots of exposures are occurring during a time of highly plastic brain. The first data release was in April 2017, and any day now, the third data release 3.0 will be done, where the full baseline data and the first half of the second wave of MRI data will be released. Now, at the end of this 12-year effort, there'll be over 25 million non-biological measurements that will have been acquired and collected across all the participants. Each participant will have 5 MRI scanning sessions with 12 distinct different MRI acquisitions. That equals over 700,000 MRI scans. And the data are, they are very strictly regulated to with regard to protected health information, in particular because of the exposure of this particular data set to the public. And that is mostly secured to the National Data Archive. Right now, currently, we're releasing demographic, behavioral, and raw imaging data in real time in the form of these fast track releases. And the data core, which I'm a part of, provide these yearly updates of condensed, tabulated data for analysis and easy access, as well as some minimally preprocessed data sets for the imaging, where we're doing very vanilla stuff like distortion correction, bias field correction, motion correction, things like that, so that people can take the data and do their favorite ways of processing the data as well. There's also this a data exploration portal called DEEP that is used to assist with the tabulated data and doing the proper statistics and multivariate analysis and selecting the correct variables and things of this nature. That's easy access. But for our community-derived measures, for stuff that you or I or anybody else has developed that want to share with other people, the only real mechanisms to do that right now is through the NDA, after you've signed your specific data use agreements. And so what we've been working on, and it's been a soft release for several months now, and the paper on this is should be going out and be available any day now. But we've been working on a secondary data share called the ABCD 3165 and the ABCC analytic utilities that are designed to supplement the data sharing with an accessible platform for the community of shared derived data, assist outside investigators that are not me or other ABCD investigators to conduct into the state-of-the-art statistical analysis for using several software packages that were designed for ABCD and ABCC, and provide kind of an integrated data analytic utilities for kind of results verification, which is related to the replication problem, which I'll be talking about a little bit later. I put a few links on here so people can do some more reading about some of the details and where to find some of the utilities that you can see below. Now, what this share contains is community-derived BIDS derivatives, so analysis-ready type data. I'll talk briefly about what that means, as well as the ABCD BIDS pipelines with very specific select free surfer and cortical thickness and other types of measurements. And there's several other data elements that people would be very interested in with regard to diffusion imaging, individualized networks we'll talk about, the task fMRI prep, all of that is being uploaded right now and will be released in the next few months. But what I thought I would do today is go over a few of the items in here, just so you can get a sense of what's available and that can help you facilitate your own work. And the ABCD BIDS pipelines, the ARMS, which is the ABCD reproducible match samples, and SEND, which is a sandwich estimator for neuroimaging data. And I'll talk about that now. So again, just highlighting the repositories where you can find some of this stuff if you're at all interested for your own work in using the ABCD data. And okay, so the ABCD BIDS pipeline is containerized software that has very standardized inputs, very minimalistic interface that's built on the human connectome. The human connectome pipelines and utilities there. It kind of uses the best in class of the software utilities, and it's all containerized to make it really simple to use. There are many additional options available, but the pipeline can be run by simply providing the path to the inputs and the outputs that are in BIDS format, along with the free surfer license. If you want to use the defaults, and that's about it to, in essence, run and get the outputs for some of the analytic utilities we'll be talking about shortly. Now, importantly, particularly for those collecting clinically rich samples, it not only works with ABCD data, but also with legacy and non-ABCD data acquisitions and specialized populations, including those with aging, who often have increased ventricles and increased size ventricles and things like that, which make it difficult often to handle the data. Again, it's utilized this BIDS folder format, and there's tools to help you reorganize your data into this similar format, but standardized that allows for easy data sharing. We won't go over the details of that, but this is the basis of which it's built. Now, the derivatives, the BIDS derivatives, are simply the outputs of a given application. The standards are not complete, but we work closely with the governance body for them to help move along and build new standards for new types of derivatives. But importantly, if you're interested in using these data, the downloads from the NDA can be data type specific and really assist, because of the general size of how much data exists, facilitating usage and reducing costs, particularly on your own servers. There's QC, or quality control, is critical. So there's several pieces of information with regard to the quality of the data that you'll be looking at. And there's also what we call its executive summary, which allows you to set eyes on the data in a very easy way to make sure that your data that you're using in your analyses are good to go. So just to conclude this piece here is that this pipeline is now available. The data for at least the baseline is mostly available, at least for rest and structural. Of the almost 12,000 subjects scanned, about 10,600 have enough data to be in this particular data resource. So if you have any more questions on that, feel free to contact me or my team. Now, the handling these data after you're done, even after you've downloaded it, can be quite difficult, and simply because of the size and number of participants being used. So if we're doing, for example, if we're doing some type of brain-wide association study, that often has to take into account many covariates that impact the analytics. And the ABCD study in particular contains lots of covariates that are normally independent, but are actually now nested. And I provided, I say an example here just so people know what I mean, because this causes some problems with some of the statistics of these data. So if you have, if we have 21 different sites, MRI data are acquired across 3 different platforms, GE, Siemens, and Philips. And as a result, the site and platform effects have effects on the impacted measures and need to be controlled for. But because not all of the dependent measures are overlapping, that means that not every site has a different scanner platform. The covariance structure of the data means that it's nested. And that can be problematic. So this nested covariance structure can be accounted for with our typical linear mixed effects models. But because of the imaging data, there's tens of thousands of data elements and thousands of subjects with multiple repeated measures. It can be over 1 billion data points, and properly adjusting, which makes it hard for typical linear mixed effects models because of they're not as efficient. And properly adjusting for statistical significance, like in the case if you want to do permutation testing or something of that nature, they become less reliable as you have more and more nested variables and covariates. And so the marginal model approach was developed to allow you to test brain behavior associations, but control for all the nested and covariance structure in a very efficient way. And that is in essence this tool called SEND is also available for folks for their types of analysis they want to use. Now it's very basic. Again, because of timing, I'm not going to go through all the details, but it simply is you generate a model that you can test on your specific sample. My cursor's not working, so I'm having trouble pointing, but you can, if you're interested in looking at specific types of networks effects, you can do a specific type of a testing enrichment, which looks at specific networks effects on whatever kind of question of interest you're looking at. But you also can do the types of permutation testing. In this case, it's called a wild bootstrap, to make sure that your statistical significance of what you're seeing is actually accurate. And that can be done both at the box-wise level and at the network level. At the bottom here in the panel C, it just gives you just a brief example of the efficiency of this, such that if you want to do it in a sample that's as large as ABCD, you can do it very efficiently with this type of model relative to the traditional mixed effects model that we are all used to seeing. So reproducibility, this is gonna be a major topic of the rest of the talk. And I think there's a little bit of this is why I call it, this a turning point in our field, is that there's been lots of papers that have slowly trickled, but are rising in nature about the ability for the results, many of the results in the literature that we see to actually be able to be reproduced. Some of that can be based on simply decision-making and how we're processing specific data or handling the data after it's collected. That's what this paper is about. But other aspects are simply because of things like sampling variability, which we'll get into a bit later. But to handle this, we've generated these matched samples called ARMS in ABCD, which allow people who are interested in these data to actually, to actually kind of replicate their specific findings within the data set themselves. And what this is it's very simple, where we've matched and provided some tools to generate matched independent groups across many demographics, family structure, twin status, things of this nature, so that if you can find a finding in 5,000, or in half the sample, that you can replicate it and know what type of reliability that you should be expecting for any given result. This is very important because lots of the results that we'll see very shortly or lots of the things that we're searching for, particularly in brain imaging, often have very small effect sizes. And if the sample size is not large enough, you can get a result, even though it won't actually really replicate in an independent sample. So this we think is really important, and we've generated these match samples to facilitate folks when they're using these data to make sure their results are replicating. The good news is, if we look at, of the MRI data, is if you actually look at these different match samples, we'll call them ABCD1 and ABCD2. This is just looking at resting state functional connectivity and cortical thickness. But if you look at them independently, this is just an average of typical connectivity on the left, and the variance structure across the participants, they're almost the point estimates are almost perfect, meaning that they're highly reliable and very reliable data on average. And that is for both the functional connectivity and cortical thickness, which we think is very promising. And it also highlights that these matched samples are the data associated with these matched samples are also highly similar. Now, but I'm going to give you just a quick example of how you might want to use, like a vignette, in essence, of how one might use these matched samples with some of the tools that I've been talking about to look at a brain-wide association study. Okay, so in here, what I'm going to do is, in this prior paper from a couple of years ago by Wes Thompson, what he did was a kind of sophisticated family structure PCA analysis across all of the cognitive measures in the data collection in the study. And what he was able to find was that these 3 specific traits were identified: general ability, executive function, and working memory. And this is across the first wave of the ABCD data sets. We've now replicated this particular finding in our specific match samples. Okay, so we have measures on every subject of general ability, executive function, and working memory. And now what we can do is we can say, well, what is the connectivity, for example, in the brain that relates to each one of these specific measures? Okay, and then we can also ask ourselves, how well does that particular finding replicate in an independent sample? Okay. All right, so this is what we're looking at here. On the top, what you can see is the relationship between, this is just for general ability, but the relationship between general ability and each cell in the connectivity matrix for both these 2 independent data sets. Now, the good news about this is that, although it's not labeled properly in this figure, I apologize for that, is that an R-squared, you can reliably detect the same results with an R-squared of about 0.67 across these 2 matrices, which is relatively strong if you use all the subjects in both ABCD1 and ABCD2, okay? Meaning that some of the findings in here, I'm not going to go over the specifics, but are highly reliable and replicable across the 2 data sets. The bad news is if you now use one of the data sets as the baseline, in this case ABCD2, and now we subsample in a permuted way different numbers of participants from 50, 100, 150, 300, 400, 500 participants, and run the same analysis and the same reliability analysis that we see in A and B. What you can see in D is that in order to reach 80% reliability, which is the dotted line over there of the max, which is that 0.67, you need almost 800 participants to be sure that you're getting a reliable result. That is a very large sample, much larger than we actually ever truly anticipated. And that is with subjects in ABCD that have at least 10 minutes of resting state fMRI data. If you look at subjects with 5 minutes of resting state MRI data, which is an E, right? So now it's the same subjects, but we just reduce the number of data used to generate their matrix. What you can see is because the reliability of the fMRI measures gets worse, that the number of participants that you need to reach that 80% reliability jumps by almost 500. So again, this is beginning to be a little bit scary, meaning that the sample sizes that we think that we need to do these types of brain behavior correlations are much higher than we ever anticipated. And the story doesn't really change as if you look at some of these other measures, like executive function and learning ability. In fact, it's a little bit worse because the reliability of the behavioral measure is worse in some of these other ones. But again, it's beginning to highlight a trend. So just to conclude this section here is that the data look good and are reliable for ABCD study. The ABCC, the Data NDA Collection 3165, is providing an additional and growing resource for the community to conduct analyses and actually share their own derivatives of ABCD data with the community. But despite all the efforts in maximizing the signal, data harmonization, due to the remaining noise and likely sampling variability, which we'll go over shortly, the number of participants that are required to get reliable findings are going to likely be very large, much larger than we we ever anticipated.
Damien, can I just jump in with a quick question?
Sure.
On your correlation, and I'm not sure if I have the variables correct, but was there a difference in terms of the replicability of cortical thickness versus the connectivity measure?
We'll go into that a little bit in this next part. There is a slight difference, and it's actually it's for the brain behavior relationships. It's actually a little bit worse. And the reason for that is a couple-fold, and I'll get to go over in a second. Is that the reliability of the cortical thickness measurement itself within subject is actually quite high, but its association with these, with the strength of the association with these, with the behavior is much lower. So the reliability of the brain behavior relationship is lower than the functional activity, and I'll show you some of that in a second. Even though the reliability of the measurement itself is higher, much higher.
Thank you.
Yep. Okay. So now we'll go on to this replication problem, and it really is at a turning point, and really how we need to be thinking about, really thinking deeply about how we conduct a lot of our work. Okay, so this is a great example of team science. It's a paper, you can get it already, it's under review, but it's available for people to read on bioRxiv, of just an amazing effort by a student and a postdoc, Scott Merrick and Brendan Turbo-Clemens. My laboratory providing a lot of the data, and Nico Dozenbach's laboratory providing much of the analyses. But it really is an amazing team effort, and kind of a way that we're going to have to, start doing a lot of our science to really conduct really strong experiments across with data sets this large and with and this complicated. All right, so here we're going to just go through.
Damien,
yes.
Just want to jump in again, and don't answer it if it's going to derail you, but the ABCD study was initiated to ascertain a large cohort of youth and to sort of map their trajectories both cognitively and neurobiologically. But in order to do that, you needed a huge sample, which required many sites, which introduced all the methodologic complexity of what applications you're using, how to establish reliability, and reduce whatever methodologic noise variance. But what you focused on up until now is all this methodologic stuff you had to go through to enable you to be able to do the primary aim of the project. Is that an accurate characterization?
Well, I'm not trying to figure out what's the characterization.
I'm saying that the primary goal of the study wasn't the methodology. That was something you had to do in order to carry out the aim of the study.
Oh, yeah, the primary aim of the study is certainly not the methodology. But what it's unveiling is, and this is not really about the methods as much either. It's about, which you'll see here shortly. It's about how we think about how we do our studies, which is related to ABCD, because already there's lots of studies that are looking at, some of the main aims of the study, but probably without sample sizes large enough to get true effects. And that is in part of some of the early things that we're realizing from of just obtaining the first round of data, that we need to rethink quite a bit about how we're doing studies in general.
No, I see where you're going, and what you got into this to answer a certain question regarding these correlations, and then in order to do it, you had to ensure that the methodology was robust enough and to do it. And in determining that, you understand what was a primary source of the unreplicability problem that the literature suffers from.
That's right, and it's kind of it's very humbling, to be honest. But I'll get into some of the stuff about, particularly across multiple sites and things like that, in very short order here. So we can. So, but I think I like going over this part of the talk, in part because it really is just this fundamental. It's just a fundamental statistical thing that we all know and have all heard of, but it's kind of, now it's real world. Now that we have this data set that's large enough for us to test this stuff, that it really kind of really, there's some examples we can show that really makes it hit home, I think. So the questions here are simply, the first thing is, well, what are the effect sizes of the brain behavior correlations that we should be expecting in our data? Okay, so to do this, for this sample, what we did is a couple ways. I'm going to show you the data from the highly denoised data, and like the clean, pristine data. It's about 4,000 participants of the total, but all the stuff I'm showing here replicates in the full sample. And the idea here is to more precisely estimate the effect sizes of the correlations. And then similar to what I described earlier, through these resampling techniques, examining the effect of sample size on reproducibility. Now, this was done across all, I can't tell you how many different ways, but with cortical thickness and connectivity, looking at vertices, ROIs, networks, and looking across many different variables, demographic variables, cognitive variables, other mental health variables, and just trying to get a real sense of what the expectations should be across the samples. So the first thing here is that if you look across all this stuff, right, that we look at the distributions, in the left is cortical thickness, and the right is functional connectivity. The largest univariate, if we just do univariate tests on the data, effect size was an R of 0.16, okay? And you can see the top 1% and the median overall effect size. The point here, regardless of we're looking at connectivity or structure, right, is that the effect sizes are very small, okay? Likely smaller, definitely smaller than we're used to seeing in literature, and we'll explain probably why here in a second. So the question is, the next question is, does the reliance on typical neuroimaging sample sizes, ends of 25, 50, 60, 100, provide a parsimonious explanation for why there may be replication failures in the literature, and if so, why? So here is we have to think very, very clearly about what sampling variability is and how much it means. So sampling variability is simply how much an effect size estimate varies between subsamples of a population. It is objectively boring. We've heard it all in our statistics courses, but we don't often consider it when we're doing a lot of our work and designing some of our studies. And so just as this example really kind of hits home for most folks. So this is an example of that Scott put together of sampling variability in the exact sample that we're looking at with the brain, but just looking at the relationship between age and height in the ABCD sample. Okay, so I might pull out a sample of 25 subjects, and I can get a reasonable R. Something you'd probably expect, where you see an R of 0.85. That's actually maybe relatively high. Between 108 and between 9 and 10, where you see height does have relationship with the moderate effect size between 9 and 10 years old. But I might also pull out a sample of that same population, and I might randomly get a sample that has an R of 0, where there's no relationship between age and height. Okay. Now, if I can sample over and over and over and over and over again, I'll probably get a distribution that looks like this on the left. Well, I will get a distribution that looks like this on the left. This is actually real data, where in some samples I'll pull out, I'll get a ridiculously high relationship where it almost reaches 1. In other samples, regardless of how small, I might even see a negative relationship between height and age. Okay. Now, and that's with a sample of 25. Now, only if you now go out and you start collecting more and more and more and more data, do you reduce the sampling variability such that you can get an approximate true effect size, which is just right around an R of 0.5 between these ages, of the relationship between height and age. If you have much smaller sample sizes, you increase the probability that you'll get an outlier or an event that outliers along this distribution that is not properly reflective of the true effect. And this is a good example because we feel fairly confident about the precision of our measurements with height and age relative to some of the other measurements we do with MRI. Okay. Now, the important thing here is that the only way to get rid of the the issue to sampling variability is to increase the sample size because it is just a random source. It's just a random source of error. And this is very different than systematic sources of error, where methods variability, p-hacking or publication bias. In this case, right? Typically, even if we had 100 different investigators study the same question, the only ones that will get published are typically the ones that have a high effect and a significant result, which is likely very much inflating the effect size that you see in the literature. And so when we do our power analyses based on literature and things like that, they're probably inaccurate because of the publication bias. And other things like head motion and acquisitions and different sites and other kind of systemic sources of error that actually can be corrected, and we'll talk about some more of that too. And so what do small effect sizes and sampling variability mean for small sample studies? Well, what it means is that, and this is the example, this is just some plucked, examples that you can see of looking at specific relationships with cortical thickness on the left and psychopathology or cognitive ability and functional connectivity on the right. But with small sample size, you can get highly significant results, right, that actually go in completely opposite directions, depending on, in this case, depending on what sample you just randomly pulled from the population. Okay, so our studies that are small, when we see this in the literature, and we often do, where you see results going in the opposite direction, this is likely why. So the observed effect sizes suggest consortium level data are required to reproducibly detect univariate brain phenotype associations. And in our case, to achieve 80% power, at least in what we examined here. This is not for everything, of course. You need almost upward to 2,000 subjects, which is, again, very humbling. So the question, of course, this is sort of brought up earlier by Jeff, but not to its full extent, but the question is, well, ABCD is multi-site, increases lots of noise because you have multiple different places that are, providing data. Is that the reason why we're seeing what we're seeing? The answer to that is likely no. So this is the comparison of what I just showed you, but using the human connectome data in adults. And in essence, even though the HCP sample is much smaller, up to the point that you get almost identical distributions. The results are almost identical. And that was all collected on one scanner. And the same thing goes if you even take one of the larger samples from one site, even within ABCD, is that the distributions are largely the same. And so this is very, very unlikely to be just a phenomena of being a multi-site study. Now, multivariate and machine learning methods are often used today. And the question is then, do those improve replicability? The answer to that is they actually do to some extent, but not probably to the extent we would like them to. So again, on the left is cortical thickness, on the right is connectivity. Again, I apologize because I'm not able to point to, but I'm just going to try to walk you through it with my words. But on the y-axis is the discovery results of this is now the ability to, this is like using cross-validation or typical ways that we do, a lot of the ways that we do typical multivariate machine learning analyses, where you were looking to see how, what's the correlation of the true outcome relative to the predicted, right? And that's what's on the, and then the x-axis then is actually building the model and the sample and testing it in the replication cohort. What you can see is that in the discovery sample you get all types of variation of like your ability to predict a specific outcome that is highly dependent on sample size. But a lot of that is the replication or ability to replicate is for many measures, particularly for cortical thickness, is quite low. Almost to zero in some cases. Although there is, even though it's much lower, there is replicability. The connectivity is a very similar story, although it's a little bit stronger with a lot of the other measures. So this is related to what I was talking about earlier, Jeff. Okay, so does the reliance on this typical neuroimaging sample sizes provide an explanation for why we're seeing replication failures in brain-wide association studies? The answer is yes. Small effect sizes with sampling variability produces these statistical errors that undermine their ability to reproduce findings. And there, if you are really interested in digging in and learning kind of the more, really digging into details, is a nice review by Anthony Dick and Wes Thompson that from ABCD that really goes into details about the issues I've been talking about here as well. So that again is very humbling, and it makes you have to rethink a little bit about how we're doing some of our population studies. But then the question is, well, what can we do about it? And one of the ways is that some of that systematic noise, I feel like we feel like we can get rid of to get us to more manageable types of studies if we really think strongly about it. And that's just increasing the signal to noise, potentially using individualized networks. And even in ABCD, we're beginning to explore this a little bit. So there are other solutions, that we'll probably need to deal with. So, we want to improve standardization of a lot of the ways that we're procedures of data collection of behavior and clinical data and MR data and other physiologic measures, so that we can combine data more easily, increase the N. But again, this is more about optimizing the signal to noise by reducing some of the systematic error in our samples. All right, so it seems like forever ago, but one of the things that I kind of kicked off my career with was using graph theory to identify networks in the brain using rest and state functional connectivity. Here, in this case, we're simply the nodes here are vertices or regions of interest. The connections between them this is functional connectivity. And we use things like community detection which kind of maximize the intra-community edges versus the inter-community edges to identify specific networks in the brain. That work, again, it seems like forever ago, but it's actually not too long ago, has exploded in recent years with regard to all the different techniques and ways that we can characterize and identify specific networks in the brain. Now, most of that work in the past has been done by first kind of averaging all the data in our sample. So we get a sample, we average all the data together, and then we identify where the networks are. And part of that was required because we needed to increase the signal to noise, and that gave us these, relatively well-known systems in the brain. There's a small number, somewhere probably between 15 to 20 networks in the brain that are important for supporting various types of cognitive phenomena. But the problem with measuring everything based on the average of a bunch of people in the population is that nobody's average, right? Nobody's average with regard to how their brain likely looks or their specific topology, right? Now, this is kind of a fun joke. I actually was texting Grow yesterday to make sure he was going to be here today, but I can give Jonathan and Grow a little bit of flack in this talk because they're close colleagues, as well as Rachel. But what we've probably in the most recent years is really like the last few years has been a high push to really try to understand how all individuals are really specific in how their networks are organized across the brain. This work started off with a paper by Tim Lauman back in 2015 looking at the poldrome. This is Russ Poldrack, a colleague who scanned himself like a madman for twice a week for about 10 months, and had so much data in himself that he reduced the signal to noise so much that you can see very specifically exactly where his systems existed in the brain. Now, this was followed up by what's called the Midnight Scan Club data, where 10 individuals, because you can get scanning very cheaply in Midnight, it only costs about $25 an hour. They said he replicated that in many more subjects and identified the same thing, where you can see lots of the same principles of general features of what you see in the average, but there's lots of very specific topological phenomena that exist within the individual that cannot be captured by looking at the average. And this is just an example of how these systems kind of map on the traditional task fMRI. They very much closely, the match onto what you expect to be activated, even within subject. So the problem with, identifying this community structure within individuals primarily is that it requires tons of data, right? So for these prior papers, in order to reach really strong reliability within the subject, you needed upwards of 40 to 60 minutes of clean MRI data, which for most of our studies is likely not possible to do for a lot of the questions that we want to ask. So for ABCD, I tried as hard as I could to get them to collect 40 to 60 minutes of resting state fMRI data in every subject in the 10,000 person childhood study. I didn't win, but we were able to get to the point where we collect 4 to 5 minutes, 5-minute resting runs, about 20 minutes of data, trying to get 10 to 12 and a half minutes of good data per subject. We didn't quite get there, but we're reasonably close, which is in essence the 80% mark on the figure in B. It's low, but it's better than it could be. So one of the things that we've been working on with Steve Peterson's group at WashU is some other methods more that are outside of community detection that are more supervised to be able to identify these networks with less data. Okay. Now this example is called template matching. There's a couple of papers on it now where we have a specific network where we have a template network, a group average network where we can identify specific seeds per network. This example is the default network. We then can generate from those seeds, we can generate an average kind of map of what a given correlation structure of a given network might look like. And from there, what you do is pretty simple. You just, for every single voxel, you measure the connectivity pattern that exists in there, and you compare it to all the different networks with the eta squared measurement, the network that it looks most closely like. It gets assigned to, and if you do that for every system, you can identify these individual networks, hopefully, presumably with a little bit less data in a much more supervised way. So what we've been doing is we've been validating that this can mimic, some of the gold standards and determine the amount of usable data necessary to actually get these individualized networks. So the short of it is and this is using the Midnight Scan Club data to do this. It's pretty simple. You can generate, split, individual subjects data in half, about 2 hours of 2 hours each, where you can do the very similar type of subsampling and just compare different methods and different time allowed for each, for when you're generating these systems. So in this case, and then of course you can use what's called mutual information to compare the network structure to see if it's actually still individualized or not. So you can compare community detection, which is the gold standard, to community detection, or you can compare community detection to template matching, or template matching to template matching, or and so on and so forth. So this is just a picture of looking at what the community structure looks like in using community detection in one half and template matching in another, and I give it in a specific individual. And what's nice here is, as you can see qualitatively, is that some of the very unique specific structures in the system seems to be replicated when you're using, using both methods, even though it may not be perfect. Okay, we can actually quantify this with this mutual information. So here we're looking at looking at my network versus my network, like self versus other of these two different split halves. And the point of this graph is simply that the distributions are split, meaning that you can very clearly determine who's who based on their network structure across the two data sets. And this is doing the same thing, but now comparing community detection to template matching, and you get a very similar split of the distributions, suggesting that template matching is mimicking, to a close extent, what you kind of expect, or the level of replicability you get with community detection, even though potentially using much less data. And here's just showing that, so now the question is, how much data do I need to get to that point? So it's very similar to what we talked about before, but instead of subsampling subjects, now we're subsampling time. And what we can show here now is that if I take my baseline of template matching, and then I take a subsample minutes, the other subjects, the other data, I can see 10 minutes, 20 minutes. How much data do I need? The longer the story, the short of the story is that you can start reaching kind of maximum reliability at a much slower level, somewhere between 10 to 15 minutes. You starting to see some plateaus of using this particular technique, which is highlighting that we can potentially use this more supervised technique to get individualized systems in samples that have much less data, like ABCD. Now here's just a picture of looking at how the individual topology exists across subjects. In just 200 subjects in ABCD. It's not been run on everybody. And again, what you can see is that there's lots of similarities across general similarities across participants, lots of variability of the topological organization. Okay. Now, a lot of studies don't even have 10 minutes of data or 20 minutes of data to be able to generate these individualized networks. But we think that we can leverage what we're learning from ABCD to assist in reducing the noise in other data sets for these brain-wide association studies in mental health. So to do that, what we can do is, even though any given voxel may not be, may not be part of a given network for every subject, you can generate the probability that it is in a given subject. Okay, so if I generate the probability that a given part of the brain is in this specific system within a given individual, I have some confidence, very similar to what we often do with volumetric structural MRI. You can measure with some confidence that that system is going to be really representative of the system and not for any given person, and not necessarily a combination of multiple systems, which we think can increase the noise. And I'll show you why we think that. So here is just a quick view showing that if you use our split samples, our ABCD1 and ABCD2, and this is just with the frontal parietal network, but it's very similar results elsewhere, is that these probabilistic maps are highly reproducible, that is looking from A to D, and they also are highly related from method to method. So even though the number of participants or the amount of data to get strong info map communities is not sufficient, we still get high relatability across the different methods. But the point here is that it's highly reliable both in the surface, in the cortex, but also in the subcortical structures, which we're often interested in psychiatry and mental health. Now, what we think we can do is now we can leverage this data to generate new types of parcellations that are based on the probability that a given signal is in a given parcel. And we think that for data population studies that they may help in decreasing the noise, increasing the signal, and allowing us to have smaller samples. So here is just two, what I'm showing at the top is an 80% probability. It's generating a parcellation set where there's 80% probability that that given parcel is in that given network for a given subject. On the bottom is what we typically use, is what we often use with the Gordon parcellation, which is very, very, very well known and often used for many, many types of studies. But you also can see the signals in the matrices on the right is that the within network, which is the boxes along the diagonal, and the between network signals, which is the boxes on the outside of the diagonal, seem to have a little bit more. They seem to be more crisper, a little bit have more signal if you use these probabilistic parcellations relative to use the typical kind of Gordon type parcellations that we often use in our stuff. Okay, now this is just a qualitative look, but we can also look at this quantitatively. This is examining all the networks. The dashed lines here are the probabilistic parcellations, and the solid is the Gordon. But to make a long story short, that the relationship that I'm showing qualitatively exists quantitatively as well, and that's what that is showing. All right, so we're coming to the end here, but I just want to highlight why we think this is important, and why I'm doing kind of a methods talk instead of my typical talk on actual findings. I think it's so important for our field. So these are the slides I showed you, just the slide I showed you earlier about the brain behavior relationships and how many subjects you need to be able to get a reliable finding, looking at in ABCD, looking at these general ability, executive function, and memory. Okay. This is what the same curves look like when we're switching, instead of using the Gordon parcellation to using these probabilistic parcellations that I just described, that increase the reliability within parcel. And the point here is just to show is that for some measurements, for some comparisons, it can have quite a profound effect. Others not as much. But just as an example, and this is just one specific test that we've done to increase the signal to noise in our data, is that if you want to reach the same reliability with the probabilistic parcellation relative to the Gordon, that can be quite highly significant in some of our comparisons. So in this case, like in the general ability, it's maybe not so much. You save about the same reliability can be accomplished with about 200 less subjects. But in examining executive functions and learning memory, the same reliability can be accomplished with about 400 subjects fewer. And that is an enormous benefit. Now we have a long way to go on this. This is hot off the presses. We're still working deeply on getting the details on this, but this is just highlighting some of the gain that we think that we can make by thinking deeply about how we're doing our analytics, such that we're maximizing the signal to noise, increasing the effect sizes, and thus reducing the amount of sample we need to answer tough questions. So I'm going to end there, just highlighting that these traditional approaches of individual network identification might be difficult outside of subjects with large amounts of movement-free data, but it may be possible with other approaches like template matching that we've highlighted with ABCD, and that individual topological characterization or consideration when we're doing our analyses are likely going to help with getting more reliable findings. Now, all subjects in ABCD are being released with their personalized network, so people can play with it however they want, and looking at outcomes. That should be out in May of 2021 via the ABCC that we talked about earlier. And then for some types of questions, generating region sets that utilize this individual topological information, and in our case, these probabilistic parcellations have an ability to reduce sample sizes to identify meaningful and replicable effects. So I'm gonna end there. I hope you got a sense of some of the main goals for folks that are in this field. The new ABCD BIDS community collection. Hope we learned about highlighting the replication problem and things we need to think about and how we do our studies. And a few things that we might be thinking about and directions we should go and what we should be doing about it, increasing our SNR. Again, thanks to all of my lab and various collaborators. It's been a wild ride, and of course, all the funding that has been acquired to conduct all this work, particularly the funding related to ABCD. Thank you all.
Thanks so much, Damien, for a great talk. I'm not sure we can give you a round of applause, but I'll give you one. It was super interesting stuff, and it generated a lot of attention and questions. I have a lot of questions myself, but to make sure that I go through the questions and comments, I'll start with others. First, Gaurav Patel, probably people saw this, but he wanted to mention that the ABCD BIDS pipelines are installed and available to use for NISP MRI users through the MRI unit. And this has been a collaboration between Gaurav and Damien and the MRI unit. So thanks to both of you.
Yeah, exactly. I would just highlight that hopefully there'll be some updates before too long where we'll have some bridges between that pipeline and fMRI prep, which we know is very popular too. So that's another part of the collaboration that we're working on with Columbia and Grow. So,
yeah. And this is obviously super important work for all of the reasons that you've been mentioning. So we have a question from Cristiane Duarte, which is, could you please comment on sample selection? Wouldn't you be able to also get true estimates in a sample that represents the true populations, even if the sample is not so large?
Yeah, that's the difficulty for is no matter what sample that you choose, there's always going to be some level of sampling variability, and for the types of for specific types of questions. Now, if if you're selecting very specific samples that you don't care that they relate to the larger population, or you don't care that they relate to the general population, then that maybe that's it just depends. I think that maybe it's something that's related to the question. But if you expect the findings to replicate or relate to the general population or your sample population, your clinical population, then the sampling variability is always going to be there. How much is still a question, but it's always going to be something we have to overcome. And it's, of course, sampling variability, if the effect sizes are large, the larger the effect size of the given effect, right, then then you don't need as much samples, but that will always be there.
So a related question from Brielle Schneck. Is the findings presented all focus on relatively agnostic rest and state networks that may or may not have strong relationships to particular cognitive phenotypes? Would you care to comment on whether the sample size reliability requirements would be different for task-based approaches or machine learning-based approaches focusing on identifying a specific targeted behavior? I guess you did talk about the multivariate methods.
So couple questions, and it's funny. I've given this talk a couple times now, and usually, if you're a task fMRI person, you're like very excited because remember, when we're doing these population studies with rest and structural and all that kind of stuff, right? It's typically it's across the sample, right? It's not within subject. Task fMRI in general for task contrasts, not for all of them, but it depends, typically have much larger effect sizes because it's within subject, right? You're looking at the changes of function within subject. So that those repeated measures actually increase the power significantly for task contrast, so you don't need, in fact, there's a paper that will be coming out with ABCD in Nature Neuroscience June that kind of highlights that point, that the sample size required for task contrast is much lower. But if you want to relate that contrast to some outside behavior, like a mental health issue or group comparisons of ADHD controlled or something like that, then it falls under, it's the same, and then it has the same issue. If that makes sense.
Yeah, that makes perfect sense. So I'm going to follow up with my own question, which is related to these two things. And I think that you did a great job in qualifying, that all of what you're talking about is, making inferences about the general population and not so much necessarily specific populations. But it seems like, to me, like my fear when I hear you talk about this, which is, obviously a very important finding and one that might have important implications to funding and, that NIH and others are going to look very carefully at, is that this is, like you're saying, something that is related to discovering neural phenotypes associated with inter-individual variability in the general population. But, so what I have in the back of my mind is for instance, the PET literature has in very small samples, very well replicated findings, a lot better sometimes than the fMRI literature. So you have studies that are in 15 people with a a pharmacological challenge and a PET measure, and correlating that with severity in psychopathology in a population where you have a lot of variance, like you have, for instance, in psychosis, which is my area, from people who are not psychotic to people who are pretty severely psychotic. And that in a study of 15 people, that is replicated, and it has been shown to be replicated. So to me, the kind of comment / question is, how do we think about these two ends of like more like big data and findings that are kind of representative of the general population versus other findings that might be more specific about certain populations or certain psychopathology that might still be, very informative and very replicable? And how do we kind of differentiate the two?
Yeah, I think it's a great question. Now, the question, the topics you're identifying in PET, there's a couple of things that I don't know. It's all about the effect size of the relationship, right? So if the effect size is that big with whatever you're looking at, then it's okay to have sample size of 15. It's just with the types of measurements that we're examining and the types of phenomena that we're looking at, the effect sizes are not that big. They're small, meaning you have to have large samples to be able to overcome that, or you'll be fooled by a given result, if that makes sense.
Yeah.
The other thing that you have to be careful about with regard to with small samples is it's not that they can't be replicated, right? They can. In fact, you're almost guaranteed if you have enough people doing it to be replicated. So for example, if I have and this is unfortunate the way that our system works. So if I have a sample of 25 subjects and I get a result with a given connection and executive function, let's say, right? And it's super highly significant, right? Now, number one is that the fact that it's highly significant in and itself is if the effect size is truly small, it has to be an error, because if you got the true effect size, it wouldn't be significant with that small sample. That's number one. But number two is that if you have 1,000 people that go out and examine that exact same thing, and the only things that we ever publish are things that are positive, the likelihood that someone will replicate it is actually quite high. Someone will do it and it will be replicated, but it'll still be wrong. Do you know what I mean?
Yeah.
So I think that your question about funding and like how our systems are set up is a big problem. I mean, it's essentially encouraging, because it's really hard to do this kind of thing. But I think us as scientists it's hard because it doesn't mean that everything that we do needs a huge sample. But the majority of the things that we do, the way that we're publishing now, probably do.
Guillermo, could I just add to your question? Damien, so I mean, you're in New York, and we also, our grand rounds have a reputation of being a little bit aggressive. So you'll excuse the what may be lack of courtesy. But so what Guillermo is saying is true, at least empirically, in terms of results of certain types of radioligands that are used for PET in different paradigms. But then if you then say, well, why aren't we able to detect these things with smaller sample sizes and more readily with different MRI applications? It's, as you pointed out, it's either because there's like sample variance, or the effect size and the dependent measures that you're looking at are small, or there's method variance that's being introduced. And it's a problem. And it reminds me a little bit of, the genetics, the trajectory of the way genetics has been. Initially, we had epidemiologic genetics, and then in the 80s, molecular with RFLPs were introduced, and there was like decades of spurious or unreplicable findings. And now we have a situation where GWAS looks like, everybody said with the GWAS, you need more and more numbers, more and more numbers. There was like in MRI, you'd have Tesla envy. Well, and you have population, and then Michael Wigler came along with CNVs, and you needed like 30 people, and you could detect the CNVs. So it's a methodologic challenge, and it's not that ABCD is doing anything wrong. ABCDC seems like it's been cast in the maybe enviable or unenviable position of having to, pull back the curtain on the emperor's new clothes and say, whoa, there's a lot of things that have obviously been problematic with studies previously published with lower sample sizes or methodology that we're needing to sort out to make this successful. And it's really eye-opening.
Yeah, it is. And I appreciate you're looking back to the genetics literature, because it literally is the identical problem. I mean, the same problem that they had 15, 20 years ago with common variants, which everybody now just takes, it's like everybody just knows, that this is the thing. It's identical. It's literally the same thing. So it's just now we're just recognizing it now. So and you're right. The thing with the ligands and PET, again, within subjects and looking at effects of within subject change is very different than looking across the population. So that's also a very important point. So
I think we have a little more time, so I have some more questions here. So Rachel Marsh is asking, she's curious to know your thoughts about recent findings showing poor reliability of NIH toolbox cognitive battery, and what that means for ABC data. That's one question. And a second question is that she assumes that some ABCD sites have resumed data collection post-COVID. Are those sites also assessing the impact of COVID on kids' families?
yeah, that's a really good question. Yeah, I think that there's very unlikely we're going to be changing midstream in ABCD with regard to NIH toolbox. But remember, but the reliability of our cognitive measures is extremely important for this fact too. And I tried to I had to go over, I didn't have time, so I kind of went over that fast, but was highlighting that a bit, is that's a big issue, as well, that we need to get better at more reliable, behavioral measures as too. That's a big issue. But remember, in ABCD, it's not just, we're not only, we're collecting, hundreds of thousands of variable measures and things. It's not just some of the big ones. So I think there's lots of data to be able to answer questions even outside of NIH toolbox related to cognition. As far as COVID goes, yes, sites have been able to resume scanning, and there are absolutely there's a big effort right now. I don't even know everything in my brain, because it's a big operation to assess the impact of COVID on multiple fronts. So that we're definitely utilizing the ABCD study, and there's several sub-studies that are going on related to COVID within the study right now. So that is definitely a target. And so they'll hopefully, it won't be anytime soon, but eventually the data will be released for folks to look at as well.
Thanks. And Guru Patel has a question. To what extent do you think that the small effect sizes between resonance cortical thickness and behavior is also a biological issue? For example, structural measures will only ever have little predictive power for behavior versus more functional measures. That's kind of related to what we're talking about.
Yeah, no, I think that it absolutely is related. We put a lot of stock in in some of these measures, but they're very crude, for these really complex behaviors. So I think that's part of the point. That's why, until we can figure out, that's why the effect sizes are small, in essence, right? They're small in part because the measurements that we're getting they may be important, but they're how well they kind of match up to these complex behaviors is probably limited. Either we're not doing it, we're analyzing them right, or they're just the wrong type of measurement for that type of signal. Doesn't mean it doesn't mean that they're not, they do have some relationship, or they're not meaningless. I think that's an important distinction. And I think again, going back to the genetics analogy, it's another example. In fact, the effect sizes of the brain behavior relationships is actually quite, several times larger in the brain versus what you typically see with common variants in the genetics. So I think it's it's related, but I don't think it doesn't mean it's meaningless. It just means that the effects are small.
Mm-hmm.
Yeah.
Have a few more questions. An anonymous question.
Myrna had a question. Guillermo, do you feel there was any value using hypotheses from small N studies to test in ABCD?
Yeah, that's an interesting one. Here's what I think. I've been thinking about this a little bit. Yes, I think so. Absolutely. But you have to change our thought process a little bit about how we look at results. I think, this is like I said, I'm still cogitating on this a little bit about how we think about smaller studies as essentially pilots for larger studies, and the reason is because we gotta, in order for us to be right, and to really understand what we're looking at as a potential, as a potential variant that we could maybe test later with the big sample, is that you have to get rid of the notion of p less than 0.05 as significant. And I kind of alluded to that earlier, is that the majority of the findings, even if the majority of the findings that you'll see, even if you have a small sample, will kind of fall along the line of the true effect size, which is good. The problem is if the sample is not large enough, it won't be significant. It won't be, the p will be less than 0.5, and we'll just discard it.
That's even before you correct for multiple comparisons.
Exactly. Yeah. So so you have to change the mindset a little bit about how you're looking at the data with the small sample sizes. And I don't have a really crisp answer for like step by step or whatever, for that to answer this question. But I do know that I do think there's, one of the things that we highlight in this paper is that there's a reason why we're here today, and it's because of lots of small sample studies. We're learning a lot about how we're supposed to be doing this thing, So there's definitely a place we just got to think about a little bit more deeply about how we go about doing them, it's just, or how we think about the results and things like that,
A related follow-up question to Myrna's is, what are your concerns regarding multiple comparisons that arise from multiple research groups analyzing the same large data sets like ABCD?
Oh boy, that is a good question. I had not actually thought about that. I have to think about that a little bit more. I mean, I wonder if anybody can speak. I'd be curious to hear what your thoughts are on it. I mean, if they're, yeah.
I guess the issue is that you can have kind of like the same issues that you have in small studies, but at the meta level, right? Because if you have many groups analyzing big data sets, but only reporting what they're finding in the large data set, it's kind of.
Yeah, it's the same problem. It's a little bit different. I mean, it's interesting because it's a little bit different because the analytics are independent. So I have to think about that more, but it likely is some type of problem. I think it is not a problem. I have to think a little bit harder about, the magnitude of it, particularly if you're doing with large samples and you're kind of hopefully getting down to the true effects. I just have to think about that a little bit more. But I think it's a great question. It's providing me fodder of things I need to be thinking more deeply about.
I guess one point on that is related to the two match subsamples of the ABCD, which is a very nice feature. That I guess if you can report things that only replicate at least internally within the sample, that kind of helps to some degree with this issue of multiple comparisons correction.
Yeah.
Another question that is related to a comparison with GWAS data. Sorry, this was just saying thank you, that it was very interesting.
There's one from Guillermo. Do you see the one from Amandeep Jutla?
Yeah, so in trying to characterize neural phenotypes in something like a rare genetic disease, where the sample you can get might be inherently small, what, if anything, would be the role of resonance data from our data?
Oh, this is a great question. I love it because we've been thinking deeply about this. Again, yeah, that paper by Russ Poldrack or by Tim Lauman on Russ Poldrack data was an N of 1, right? It was extremely informative, right? The thing about that's different than a lot of our population studies, it's looking at the data within subjects, and providing very deep, specific characterizations of manipulations within a subject. And though, if you think about, particularly for rare variants, this deep precision type phenotyping work, that's, I think, is going to be more fruitful approach for trying to identify or characterize brain physiology or various types of biological correlates with the variant, than trying to get a small number of people who have that rare variant and trying to look across everybody. I think that's a good example of where kind of this precision medicine, precision phenotyping type approach, I think, would be very fruitful in trying to detail out the specifics of how the biology relates to the variant.
Yeah, can I follow up on that? So,
Yeah.
you know, that using that approach, taking a, you know, Mendelian genetic disorder and then trying to characterize it neurobiologically, is a way of validating your assay, so to speak. If you find a relationship, you know, with that, then, you know, you can take, as opposed to working from these large samples and trying to find, you know, carved nature of its joints, you know, with, you know, large ends. The only thing is that if you were to take, you know, whatever the condition was, Rett syndrome, you know, Williams syndrome, Down syndrome, Fragile X, you'd have to, you know, presumably screen with a range of MR applications to determine which might be, you know, sensitive to the genetic mutation or factor. But is anybody using that approach?
I haven't seen anything. I haven't seen anything yet. But I'll just give you a wink. Wink is that I know that it's happening. People are trying. Yeah, I think that's, you know. So I haven't seen anything out there yet, but I know it's in the works.
A related question is, how do you feel about, or do you have experience doing this type of comparison of, you know, individual subjects that might have a very specific condition that are very densely, you know, imaged or very precisely imaged, compare them to a data set of other, you know, healthy, you know, densely imaged people like, you know, Russ Poldrack and others. How do you feel about this comparison of one to a data set and like kind of talking about what's abnormal in that one subject based on the rest? Is that something that you think it's doable?
I do think it's doable, but again, it's very much depends on what you're asking, like what's the question, you know. But I absolutely think it's doable, you know. But again, it's just a little bit about, it's about the specifics, you know, about what you're looking at. But I think it's, you know, if, you know, we're actually in the midst now of trying to generate some large data sets that are precision mapped, so to speak, to, you know, help us explore that very question a little bit. And there's lots of ideas, but there hasn't really been a lot of data to be able to test the ideas yet. That's what we're kind of waiting on, I think. But I think you can do it. I just, you know, like I said, it just depends on what you're actually asking.
Okay, so I think that we don't have any more questions. So thanks again so much, Damien, for a great talk. It's super interesting and important topic. And so that's all. Thanks again.
All right.
For coming.
Much appreciated all, and thanks for having me.
Bye-bye. See ya.
Bye now.